The Cambrian Explosion marks one of the most profound and captivating chapters in Earth's biological history. Occurring approximately 541 million years ago, this period witnessed an extraordinary diversification of life forms, a veritable biological revolution that introduced complexity, innovation, and the foundations of many animal phyla we recognize today. To appreciate the scale and significance of this event, we must journey back to a world starkly different from the bustling ecosystems of the present, a time when life was predominantly microscopic and largely confined to simple, single-celled organisms. For billions of years, life on Earth was modest in its ambitions. Microbes, algae, and other unicellular life forms dominated silent, shadowy oceans where complexity was scarce. Then, seemingly in the blink of geological time, the Cambrian explosion burst forth like an awakening, a sudden bloom of anatomical diversity and ecological interactions that forever changed the trajectory of life. What made this explosion of biological innovation possible? Scientists have long debated the causes, exploring a combination of environmental, genetic, and ecological factors that collectively set the stage. Elevated oxygen levels in the oceans may have played a crucial role, providing the necessary energy to sustain larger, more metabolically demanding organisms. This oxygen boost permitted the development of hard parts such as shells and exoskeletons, which not only provided protection but also new avenues for evolutionary experimentation. These mineralized structures dramatically improved the preservation of fossils, allowing us a clearer glimpse into this transformative era. Equally pivotal were genetic breakthroughs, particularly the evolution of developmental genes like Hox genes, which regulate body plans. These genetic switches enabled the construction of complex, segmented bodies with specialized limbs, sensory organs, and digestive systems. Suddenly, life was not just existing. It was diversified into myriad forms that could exploit various niches. During the Cambrian, oceans teemed with novel creatures. Among the most iconic are the trilobites, arthropods equipped with segmented exoskeletons, compound eyes, and intricate limbs that allowed them to scavenge, hunt, and navigate the seafloor with remarkable agility. Then there was Hallucigenia, a bizarre worm-like creature studded with spines and tentacles, an example of the experimentation in body structures that characterized the time. These early animals didn't just coexist, they interacted in complex ways. Predator-prey relationships became increasingly sophisticated, fueling evolutionary arms races that spurred further adaptations. The emergence of eyes, for instance, revolutionized how these creatures perceived their environment, adding layers of complexity to their behaviors. The Cambrian seafloor also saw the establishment of extensive ecosystems, with creatures engaged in burrowing, grazing, swimming, and hunting. Such ecological networks laid the groundwork for the intricate food webs that sustained life on Earth today. Moreover, the fossil record from this period, particularly from sites like the Burgess Shale in Canada and the Chengjiang in China, provides extraordinarily detailed preservation of soft-bodied organisms. These treasure troves have uncovered an array of forms that broaden our understanding of early animal life, showcasing the rich tapestry of evolutionary pathways that were explored. Importantly, the Cambrian explosion was not an instantaneous event, but a relatively quick burst spread over millions of years, a rapid bloom in comparison to the slow pace of life's prior history. This pace of change challenges traditional perceptions of evolutionary tempo and highlights how, under certain conditions, life can innovate with exceptional creativity. Though this explosion ushered in remarkable biodiversity, it also set the stage for the ecological and evolutionary dynamics that would continue through the subsequent Paleozoic era. The innovations of the Cambrian paved the way for the rise of predators, the establishment of complex marine communities, and the eventual conquest of terrestrial habitats millions of years later. In essence, the Cambrian Explosion unlocked life's first marvels: complex structures, diverse forms, and interwoven ecosystems. It represents a cornerstone in understanding how biological complexity arises from simplicity, reminding us of the wondrous ingenuity imbued within the fabric of evolution. As we reflect on this pivotal chapter, we begin to glimpse the profound forces shaping life's journey, from its humble beginnings to the extraordinary complexity that characterizes our natural world today. This exploration of the Cambrian sets a foundation for appreciating the sensory innovations and emerging behaviors that soon followed, foreshadowing the rich evolutionary drama yet to unfold. As we move beyond the astonishing emergence of diverse life forms during the Cambrian Explosion, our attention now shifts to one of nature's most remarkable breakthroughs: the development of complex sensory systems, especially the advent of eyes and the evolution of vision. This transition didn't just change how early animals perceived their surroundings, it fundamentally redefined the interactions between organisms, setting in motion evolutionary arms races and ecological complexities that resonate through time. Before eyes came into the picture, life's sensory world was rudimentary. Single-celled organisms relied on simple chemical gradients to navigate their environment, responding to light or chemicals without forming any detailed images. But the Cambrian seas were different. With an explosion of new predators and prey, the stakes of observation and detection skyrocketed, driving selection for more sophisticated sensory organs. The earliest eyes were surprisingly simple, clusters of light-sensitive cells capable only of distinguishing light from dark. Yet even this modest capability was revolutionary. The ability to discern day from night, or shadow from open water, provided vital advantages in avoiding predators or locating food. Some of the most primitive eyes likely resembled eye spots, which can still be found in certain modern-day simple animals like flatworms. From these humble beginnings, eye structures rapidly evolved, increasing complexity. Among the primordial pioneers of vision were the trilobites, those iconic, armored arthropods whose fossilized remains reveal an impressive array of compound eyes. Trilobite eyes are among the oldest known, preserved thanks to their calcite lenses that refracted light with remarkable clarity. Each eye consisted of thousands of tiny hexagonal lenses called ommatidia, each capturing a fragment of the surrounding world. Together, these provided a mosaic image, enabling the trilobite to detect movement and shapes with impressive detail. But why compound eyes? The answer lies in their remarkable adaptability. Compound eyes offer an expansive field of view and the ability to sense rapid movement, traits especially advantageous in the predator-prey interactions burgeoning during the Cambrian. An animal that could spot a predator earlier might survive, while one that could better track prey could secure a much-needed meal. Thus, once the genetic and developmental mechanisms for compound eyes appeared, they spread swiftly among many arthropods. While compound eyes were a hallmark of many Cambrian creatures, they were not the sole visual innovation. Simple camera-type eyes, featuring a single lens focusing light onto a photosensitive surface, were another evolutionary solution. These would eventually pave the way to the more complex eyes of later animals, including vertebrates. Central to these sensory advancements were primitive photoreceptor proteins, molecules that transform light into neural signals. Opsins, for example, emerged as critical components for light sensitivity. The diversification of opsin genes during this time allowed animals to detect different wavelengths of light, potentially even glimpsing colors or ultraviolet light, expanding the sensory palette far beyond mere brightness detection. Vision's impact during the Cambrian went far beyond individual survival. By enabling animals to find mates, recognize rivals, and better explore their environments, eyes spurred behavioral complexity. Movement, camouflage, courtship displays, and territoriality all gained new dimensions. The presence of such sophisticated sensory organs undoubtedly shaped evolutionary trajectories, influencing morphology, lifestyle, and even social dynamics. Interestingly, modern neuroscience reveals that alongside eyes, neural pathways became more intricate to process the influx of visual information. This necessitated a gradual expansion and specialization of the nervous system, a topic that complements our exploration, but will be examined in greater depth later. It is also intriguing to consider that the Cambrian seas were visually diverse landscapes, illuminated by sunlight filtering through water, where animals could perceive shadows, shapes, and even the flicker of others moving nearby. This vibrant sensory environment fostered a feedback loop. Better eyes led to better predators and more astute prey, which in turn selected for even finer sensory adaptations. In addition to eyes, other sensory modalities began to develop in tandem, forming a multisensory world. Mechanoreceptors, capable of detecting touch and vibrations, allowed early creatures to sense water currents or the presence of nearby organisms. Chemoreceptors enabled the detection of chemicals, precursors to the sense of smell and taste, which played crucial roles in finding food and mates. Together, these sensory innovations set the foundation for an awakening of animal awareness. Life was no longer passive and oblivious, but actively engaged with its environment in complex, nuanced ways. This awakening would enable life's continued journey towards the diverse sensory experiences exhibited by the animal kingdom today. In the next exploration, we will delve into how these perceptual advances catalyzed the emergence of active hunting strategies, spotlighting remarkable Cambrian predators like Anomalocaris. These creatures harnessed their newfound sensory prowess to become some of Earth's earliest effective hunters, forever reshaping marine ecosystems. But for now, allow yourself to imagine the ancient oceans momentarily transformed. Daylight filtering through primordial waters, creatures gliding and hunting, their newly minted eyes unveiling a vibrant, ever-shifting tapestry of life. These are the first flickers of vision, the advent of complex light perception, marking a turning point in the evolutionary saga. As we continue our journey through the extraordinary story of life before dinosaurs, we now enter a world reshaped by a groundbreaking ecological innovation: active predation. The Cambrian seas, once a place where many animals drifted passively or fed on detritus, now transformed into dynamic arenas of pursuit, capture, and survival. At the very forefront of this revolution was a captivating and formidable creature. Anomalocaris, one of the earliest true predators and an icon of Cambrian marine ecosystems. The rise of active hunting marks a major turning point in evolution. Prior to this, many animals subsisted as filter feeders, scavengers, or grazers. But with the emergence of predators capable of seeking out and overpowering other creatures, the ecological balance shifted dramatically. This shift triggered an evolutionary arms race, driving innovations in sensory perception, defensive strategies, and locomotion. Anomalocaris, whose name means abnormal shrimp, was a remarkable organism unmatched in its time. Its fossil record spans the Cambrian period, with some of the most exquisite specimens unearthed from the Burgess Shale in British Columbia and the Chengjiang fossil beds in China. Imagine a creature about 1 meter in length, huge compared to most contemporaneous animals, with a sleek, torpedo-shaped body built for swift swimming. At its front were unmistakable jointed appendages, resembling grasping limbs, adorned with spiny structures designed for capturing prey. Its body was streamlined, tapering towards a fan-like tail that gave it excellent propulsion. Along the sides, a series of lateral flaps moved in rhythmic waves, allowing precise and agile maneuvering through Cambrian waters. Anomalocaris was not just fast. It was a masterful swimmer, combining speed with control, traits essential for a top predator. What set Anomalocaris apart, anatomically and functionally, was its feeding apparatus and sensory adaptations. Its mouth was a circular structure, sometimes described as a pineapple slice or toothed wheel, composed of overlapping plates with serrated edges. This mouth could open wide and crush the exoskeletons of hard-bodied organisms, such as trilobites, some of the era's most common prey. Moreover, it possessed large compound eyes atop stalks, capable of sophisticated vision. The compound eyes contained thousands of lenses, enabling sharp, wide-angle views to detect prey in the murky Cambrian seas. These keen eyes likely gave Anomalocaris the sensory edge necessary to home in on fast-moving or cleverly camouflaged animals. As a predator, Anomalocaris hunted actively rather than passively, a profound ecological innovation. It could pursue, grasp, and process prey with a combination of sensory acuity, specialized limbs, and powerful jaws, traits unprecedented during its epoch. This behavior catalyzed significant changes in marine life, compelling prey species to develop defensive adaptations such as harder exoskeletons, spines, and escape behaviors. The rise of active hunters like Anomalocaris sparked the evolution of more varied and complex food webs, predation-pressured populations, influencing population dynamics, and the distribution of species. It also encouraged evolutionary experimentation. Animals diversified anatomically and behaviorally to survive new threats. Interestingly, for many years, Anomalocaris was misunderstood by scientists who initially thought its parts represented multiple species. The frontal appendages were seen as shrimp-like creatures, the mouth as a jellyfish, and the body as another organism. This misunderstanding highlights the strangeness of Cambrian life and reminds us how much remains to be discovered about early animals. Beyond Anomalocaris, the Cambrian Explosion saw the emergence of other active predators, albeit smaller or less specialized. Various worms, early arthropods, and mollusk-like animals began experimenting with offensive strategies, such as grasping limbs, piercing mouthparts, and enhanced sensory organs. Together, these developments transformed Cambrian ecosystems into theaters of complex interactions where survival demanded agility, cunning, and innovation. Active hunting also influenced non-predatory animals by selecting for improved defenses. A beautiful cascade of evolutionary reactions ensued. The harder shells of trilobites, the spines and claws of other arthropods, and enhanced sensory systems all echo the pervasive influence of predation pressure. In essence, the rise of active predators like Anomalocaris was more than the story of a single species. It symbolizes a seismic ecological shift that set the tone for the evolution of complex animal behavior. It introduced the energy and intensity of chase, capture, and escape into the ancient oceans, laying the groundwork for the intricate balance of ecosystems. As we reflect on this chapter, envision the Cambrian seas pulsing with swift hunters and wary prey, engaged in timeless evolutionary contests. This dynamic interplay is a powerful reminder of how life, even hundreds of millions of years ago, was vibrant and inventive. Next, we will explore how prey responded to these new threats through an array of defensive adaptations, armor, spines, and clever tactics that helped balance the marine battlefield. These evolutionary strategies ensured survival amid a rapidly changing world where the stakes had never been higher. Following the dramatic rise of active predation ushered in by creatures like Anomalocaris, the evolutionary stage was set for a fascinating arms race, one not only dominated by hunters and their refined tools for capture, but also by prey and their evolving defensive innovations. In this unfolding drama of survival, nature sculpted a variety of armor, spines, and behavioral tactics that empowered early animals to endure, evade, and sometimes outwit the merciless teeth and claws of their pursuers. The Cambrian seas became a crucible of protection and counterattack. For many prey creatures, the development of external defense mechanisms was crucial to their continued existence. One of the earliest and most widely recognized forms of such protection was the evolution of hard exoskeletons, external shells made primarily of calcium carbonate or chitin. These protective casings served as physical barriers against predation, difficult to penetrate and capable of shielding vulnerable soft tissues beneath. Among the most iconic bearers of these shell-like armors were the trilobites. Possessing calcified exoskeletons divided into segmented thoracic plates and shield-like heads, trilobites were effectively armored walkers on the seafloor. Their ability to enroll or curl into a ball tightly protecting their ventral, underside parts functioned much like the modern pillbug, presenting predators with a hard, unyielding surface rather than vulnerable flesh. This behavior was an early example of the sophisticated prey tactics developed long before vertebrates dominated the oceans. Spines and protrusions took defensive strategies to another level. Many Cambrian animals, such as hallucigenia and various arthropods, sported long, pointed spines or needle-like projections emerging from their bodies. These spines offered a dual advantage, physically deterring attacks by making the animal harder to swallow or harming a predator attempting to grasp it, and visually signaling danger to would-be attackers. In some instances, these spines evolved to be flexible yet sturdy, bending under pressure and snapping back, offering extra resilience. Beyond physical defenses, early animals began to exploit behavioral tactics to evade predation. One such strategy was burrowing, digging into sediment to avoid detection or pursuit. Some Cambrian creatures developed specialized limbs and body shapes to facilitate this subterranean escape. Burrowing not only provided shelter, but also access to new food sources below the surface, creating an ecological niche that diversified ancient marine communities. Camouflage and disruptive coloration are also believed to have roots tracing back to this period. Though direct evidence is scarce, the presence of intricate patterns on fossilized exoskeletons and the known effectiveness of such strategies today imply that some Cambrian animals employed visual trickery to blend into their surroundings or confuse predators. This form of passive defense, friendlier to stealth and surprise, would later flourish as ecosystems became ever more complex. The interplay between predator pressure and prey defense stimulated rapid morphological experimentation. For instance, armored shells might vary dramatically in thickness, shape, and ornamentation. Some species evolved spikes arranged strategically around vulnerable parts. Others developed ridged surfaces that deflected attacks. This variability reflects the intense selective pressures exerted by active hunters, driving natural selection to explore every possible advantage. Another intriguing adaptation involved the capacity for quick escape through swimming or flicking movements. Certain trilobites, equipped with well-developed appendages for swimming, could rapidly dart away when threatened, combining armor with mobility to increase survival odds. In this sense, the balance between offense and defense began to shape not only morphology, but also behavior and biomechanics. The evolutionary emergence of sensory organs complemented defensive adaptations. Early warning systems, such as sensitive antennae and mechanoreceptors, enhanced an animal's ability to detect approaching threats. These sensory inputs allowed for better-timed evasive maneuvers, coordinated movements, or retreat into protective burrows or crevices. From a broader perspective, these defensive innovations had profound effects on ecosystem dynamics. By reducing predation mortality, they allowed prey populations to stabilize and diversify. This, in turn, created more complex food webs and promoted the evolution of further specialization among both predators and prey. This web of interactions fueled an evolutionary feedback loop. Predators evolved more effective hunting tools, while prey honed stronger defenses and more clever behaviors. Such reciprocal adaptation is a cornerstone of biological evolution and was vividly alive in the Cambrian oceans. The legacy of these early defensive adaptations extends far beyond the Cambrian. Armor, spines, and evasion tactics form the evolutionary foundation upon which countless later animal lineages built ever more intricate and specialized forms of protection. From the heavy shells of mollusks to the spiny fins of fish, and even the thick armored plating of dinosaurs. The story began here. Imagining these ancient battles beneath the waves, one can almost see slow-moving but heavily armored trilobites curling protectively, spiny creatures bristling as predators close in, and swift burrowers disappearing beneath the sediment's surface. All engaged in a timeless quest to survive in a world newly shaped by predation. As we ponder these marvels of defense, we prepare to explore the masters of movement themselves, creatures like trilobites, who not only protected themselves, but also conquered their environment through swimming prowess and refined locomotion. Our journey through life's early innovations continues, revealing the intricate balance of offense and defense that propelled evolution's grand symphony. As we transition from the defensive adaptations that helped early Cambrian animals survive predation, our focus now shifts to the remarkable pioneers who mastered the art of efficient movement in their aquatic domains. Among these, trilobites emerge prominently, not merely as relics of armored prey, but as sophisticated swimmers and explorers of the ancient seafloor, true masters of motion in the primordial seas. Trilobites, a diverse group of extinct marine arthropods, thrived for over 270 million years, from the early Cambrian to the end of the Permian period. Their extensive fossil record provides detailed insights into their anatomy, locomotion, and ecological roles. While often imagined as slow, crawling creatures, many trilobites developed exceptional adaptations that enabled them to swim, glide, and even quickly burrow, showcasing a complexity in movement that set evolutionary benchmarks. One key to the trilobites' locomotion was their segmented body design, divided into 3 lobes: the central axial lobe and 2 pleural lobes on each side, which conferred flexibility and strength. This segmentation allowed precise and coordinated movements, whether crawling along the seafloor or rhythmically flapping appendages to propel themselves through the water. Their multiple pairs of articulated legs, equipped with delicate bristles also called setae, performed dual functions. They not only enabled walking, but also acted as paddles for swimming. By moving these limbs in synchronous waves, trilobites could lift off the sediment and navigate through water columns, escaping predators or seeking new foraging grounds. Some species, such as the genus Acephus, display morphological features suggesting adept swimming capabilities, including streamlined bodies that reduced water resistance. Interestingly, trilobites possessed a protective exoskeleton composed of calcite and chitin, which provided necessary armor but also presented a weight challenge for swimming. To reconcile the need for protection with mobility, many trilobites evolved streamlined body shapes and jointed limbs that maximized effective movement without compromising defense. Certain species could enroll tightly, a defensive mechanism we noted earlier, while others maintained rigid yet agile forms optimized for active swimming. Moreover, the eyes of trilobites played a supportive role in their locomotion. Their compound eyes, made of calcite lenses, afforded panoramic and acute vision to detect both prey and predators. This visual acuity was vital for coordinating movements, especially when swimming in open waters or during complex escape maneuvers. The mastery of movement also extended to behavioral strategies. Trilobites could adjust their motion depending on situational demands, crawling steadily on the seafloor to forage or swiftly swimming upward to evade threats or disperse to new habitats. In some cases, evidence suggests rhythmic nightly swimming to avoid daytime predators, an early form of diel vertical migration that persists in marine species today. Additionally, fossilized trackways known as coprolites and trace fossils reveal that trilobites left behind complex sediment imprints. These patterns tell us about their gait, speed, and interactions with terrain, sometimes indicating social behaviors like following one another or patrolling territories. Trilobites' locomotor innovations played a vital ecological role in shaping Cambrian communities. Their ability to explore varied microhabitats, escape predators, and find food contributed to their long evolutionary success and set the groundwork for locomotion strategies seen in later arthropods and marine animals. Their evolutionary legacy resonates into the modern world. Though trilobites themselves vanished in the Permian mass extinction, arthropods like lobsters, crabs, and shrimp inherit their sophisticated limb design, flexible exoskeletons, and advanced locomotion techniques. This endurance highlights the effectiveness of their approach to mastery of movement. Visualize the Cambrian oceans anew. Swarms of trilobites flick their limbs in precise harmony, gliding above the seafloor, weaving between rocks and sediment, evading the clutches of predators like Anomalocaris. Their movements, rhythmic and fluid, form patterns etched into the sedimentary pages of geological history, a testament to one of life's great innovations. Having gained mastery over movement, trilobites and their contemporaries relied on increasingly sophisticated nervous systems to control their actions and process sensory inputs. This exciting evolution unfolds next as we decode the transition from simple nerve networks to the first true brains, a step that would underpin the behaviors and survival strategies of countless animal lineages to come. As we leave behind the remarkable adaptations that enabled early animals to master movement, our exploration now ventures into the profound realm of nervous system evolution, a critical facet of life's increasing complexity during the Cambrian and beyond. Understanding how ancient nervous systems evolved from rudimentary nerve nets to early centralized brains reveals the neural foundation enabling the sophisticated behaviors, sensory processing, and survival strategies that define the animal kingdom. In the earliest multicellular animals, such as sponges, the nervous system was entirely absent or exceedingly simple. These organisms relied on passive diffusion and basic cellular responses to environmental cues, but lacked specialized neurons to coordinate movement or process sensory input. The profound leap came with the development of nerve cells, or neurons, specialized units capable of transmitting electrical and chemical signals rapidly and in coordinated patterns. Early nerve nets, seen today in primitive animals like jellyfish and comb jellies, represent one of the first organizational forms. These diffuse networks lacked a central brain but connected sensory receptors to motor cells, facilitating simple reflexive actions such as swimming contractions or withdrawal from stimuli. Although limited in complexity, such nerve nets were revolutionary, enabling an organism to respond dynamically to immediate environmental changes. During the Cambrian Explosion, increasing environmental challenges and predation pressures fostered the evolution of more organized nervous systems. Animals required quicker reaction times, improved sensory integration, and more precise motor control, driving the emergence of nerve clusters called ganglia. These ganglia acted as localized processing centers, coordinating activity in specific body regions and allowing more nuanced behaviors. Arthropods, including trilobites and early relatives, exhibit some of the earliest evidence of such neural specialization. Their segmented bodies often contained paired ganglia within each segment, enabling coordinated limb movements and complex motor patterns. This architecture was crucial for controlled crawling, swimming, and intricate prey capture or evasion maneuvers. Most notably, the gradual centralization of nervous tissue led to the formation of rudimentary brains. These early brains were collections of ganglia near the head region that integrated sensory data primarily from specialized organs like eyes and antennae. This integration permitted animals to not only react, but also to process multiple stimuli simultaneously, plan movement trajectories, and perhaps even engage in simple decision-making processes. Fossil evidence of nervous tissue itself is exceedingly rare due to its delicate nature. However, remarkable fossils from sites like the Burgess Shale have preserved neural imprints and associated soft tissues that hint at neural complexity. In creatures like Anomalocaris, the development of large, well-structured brains complements their advanced sensory and predatory adaptations. Such evidence underscores the link between neural evolution and ecological roles. The molecular underpinnings of nervous system evolution also shed light on this transition. Gene families involved in neuronal development and synaptic communication, such as those encoding neurotransmitter receptors and ion channels, expanded and diversified during this time. The emergence of these genetic tools allowed animals to build more intricate circuits and regulated nervous function with greater precision. As nervous systems became more centralized and sophisticated, behavioral repertoires expanded greatly. Early animals could exhibit complex locomotion patterns, refined predator avoidance, active hunting, mating rituals, and environmental exploration. These behaviors required memory, sensory integration, and motor coordination. In essence, the hallmarks of cognitive abilities that set animals apart from simpler life forms. Furthermore, the evolution of early brains catalyzed novel interactions within ecosystems. Predators could stalk or ambush prey with calculated movements. Prey animals could anticipate threats and coordinate group defenses. These interactions reinforced evolutionary pressures to further refine neural and sensory systems. It's worth noting that the advent of brains did not imply instantaneous intelligence. Instead, it marked a gradual accumulation of neural capabilities over millions of years, laying the groundwork for the staggering cognitive diversity we observe today, from the problem-solving capacities of cephalopods to the complex social behaviors of mammals. This evolutionary narrative invites deep reflection on the essence of nervous systems, not merely as biological wiring, but as the foundation for sensation, experience, and interaction. From modest nerve nets to early brains, Cambrian animals embarked on the neural journey that made possible the rich tapestry of animal life. As we contemplate the rise of ancient nervous systems, our next exploration will uncover innovations beneath the seafloor. How life's first burrowers transformed sedimentary habitats and developed novel behaviors that complemented their growing neural sophistication. This subterranean world offers another compelling chapter in life's unfolding adventure before the age of dinosaurs. Deep beneath the ancient Cambrian seas, beyond the visible bustle of swimming and crawling creatures, a subtle but profound revolution was unfolding within the seafloor's sedimentary layers. Life's first burrowers were quietly transforming this environment, reshaping the seafloor habitat, diversifying ecological niches, and pioneering behaviors and adaptations that would profoundly influence the evolutionary trajectory of animals. This hidden underworld, intertwined with sediment and microorganisms, became a vibrant frontier of innovation. Burrowing, the act of digging into sediment to create refuge or search for food, represents an essential evolutionary milestone. It demanded a suite of anatomical and behavioral adaptations that were quite distinct from those required for open water or surface-dwelling life. The ability to penetrate soft substrates opened new possibilities: shelter from predators, stable environmental microhabitats, and access to untapped nutrients. The earliest burrowing activity dates back to the late Precambrian and Cambrian periods. Fossilized burrows and trace fossils, known as bioturbation structures, testify to the presence of organisms that disturbed sediments by digging or tunneling. Simple worm-like animals initially created shallow, winding channels near the sediment-water interface. These incipient burrows provided protective refuges and aided in feeding strategies such as deposit feeding, where sediment particles are ingested to extract organic matter. One of the key pioneering burrowers was the humble annelid worm ancestor, which exhibited flexible, elongated bodies perfectly suited to navigating narrow sediment tunnels. These animals evolved muscular structures and bristle-like chaetae, tiny appendages that acted like anchors, to help them push through sediment grains. This anatomical innovation allowed them to control movement precisely within constrained spaces. Equally important were the behavioral adaptations associated with burrowing. These early creatures had to balance oxygen requirements with sediment penetration, as deep sediments could be oxygen poor. Some burrowers developed U-shaped tunnels that connected sediment layers to oxygen-rich water above, effectively creating their own ventilation systems. This engineering of habitats marks an extraordinary example of ecosystem engineering long before complex terrestrial animals walked the earth. Beyond protection, burrowing also opened new feeding opportunities. By disturbing sediments, burrowers helped recycle organic material, releasing nutrients back into the environment and fostering microbial communities, a foundational ecological service. Some burrowers evolved selective feeding mechanisms, distinguishing nutritious particles from inert sand or mud, showcasing the refinement of sensory perception in these hidden habitats. The Cambrian seas also revealed specialized burrowers like certain arthropods and worms that developed appendages adapted for digging, such as rake-like limbs or stout claws. These structures increased efficiency in sediment displacement, allowing deeper or wider tunnels. In some cases, burrowing became sufficiently extensive that it dramatically altered sediment structures, a substantial physical impact on the seafloor habitat known as bioirrigation. One of the most fascinating aspects of early burrowing was its influence on predator-prey dynamics. Burrowing provided a refuge from active predators prowling above the sediment surface, effectively creating a three-dimensional battlefield. Predators evolved strategies such as probing appendages or digging behavior to hunt within sediment, while prey animals enhanced their burrowing depth or constructed more complex tunnel networks for escape. This intricate interplay drove morphological and behavioral innovation in both camps. Furthermore, the presence of burrowing animals accelerated sediment mixing and oxygenation, influencing chemical exchanges at the sediment-water interface. This, in turn, improved the habitability of the benthic environment and encouraged biodiversity. By physically altering their ecosystem, these early burrowers were some of the first ecosystem engineers, establishing environmental conditions that benefited countless other species. Trace fossils, such as the famous Scolithos and Planolites burrows, provide detailed windows into this era. These preserved tunnels and trackways illustrate the varied complexity and persistence of burrowing activity. The diversity in size, shape, and depth of these structures hints at a rich array of burrowing lifestyles, from simple, linear movements to branching, interconnected tunnels. Burrowing innovations did not remain confined to the oceans. They laid behavioral and anatomical foundations that would prove invaluable millions of years later when animals first ventured onto land. The evolution of structures suited for digging and substrate manipulation in aquatic environments prefigured the adaptations needed for terrestrial burrowers, such as early amphibians and arthropods exploring soils and leaf litter. Visualize, then, the sediment at the base of ancient seas, a labyrinthine network of animal-made tunnels alive with the movements of creatures unseen yet vital. This underworld was dynamic and vibrant, supporting life cycles, predator-prey interactions, and nutrient flux, quietly sustaining and enriching the broader marine ecosystems. Having glimpsed the remarkable innovations below the seafloor, our narrative will next turn to a different but equally vital communication realm: chemical signaling. We will explore how early animals harnessed chemical cues to sense their surroundings, communicate, and coordinate behaviors. Another foundational adaptation that deepened the complexity of ancient animal life. As we delve deeper into the ancient seas, beyond the physical adaptations and movement strategies of early animals, we uncover another extraordinary evolutionary leap: chemical communication. This invisible, yet potent language became one of the earliest and most fundamental ways organisms sensed and responded to their surroundings, coordinated with one another, and shaped their behaviors long before eyesight or sound developed. Chemical communication is the use of chemical substances, signals, and cues exchanged between organisms to convey information, provoke reactions, and facilitate interactions. In the context of life's earliest epochs, this form of communication was indispensable. Unlike light or auditory signals, chemicals can permeate murky waters, travel through sediments, and be detected without intricate specialized organs, making chemical sensing essential to the survival of countless early creatures. The origins of chemical signaling reach back well before the Cambrian to single-celled organisms such as bacteria and protists. These microscopic life forms relied heavily on chemicals to navigate toward nutrients, sense danger, and cluster for collective benefits. This ancestral reliance laid the foundation for increasingly sophisticated chemical interactions as multicellular animals evolved. During the Cambrian period, as animal diversity expanded exponentially, the complexity and utility of chemical communication likewise surged. Early animals developed specialized receptor cells capable of detecting various chemical compounds dissolved in the water. These chemoreceptors allowed organisms to identify food sources, detect predators or mates, and even recognize members of their own species, thereby orchestrating behaviors critical for survival and reproduction. One striking example involves the embryonic forms of the olfactory and gustatory systems, primitive nose and taste sensors. Though not analogous to the dedicated noses or tongues of modern animals, these early chemoreceptive structures provided organisms with essential sensory input, decoding the chemical landscape. Planktonic creatures, for instance, could detect chemical cues released by predators, enabling timely evasive maneuvers. Conversely, predators could sense traces of their prey's metabolic waste or secretions, guiding their hunting efforts. This chemical dialogue established an unseen web of signals permeating ancient ecosystems. Moreover, chemical communication extends beyond just predator-prey dynamics. It played a crucial role in reproductive strategies as animals sought mates in vast, watery environments. The release of pheromones, chemical substances emitted to influence the behavior or physiology of others, facilitated mate attraction and synchronization of spawning events. This was especially important for sessile or less mobile animals, ensuring reproductive success despite their limited movement. Some Cambrian arthropods and worms likely used chemical trails as markers, guiding others toward food or safe areas, hinting at the earliest forms of social communication. This chemical mapping would have created subtle networks of interaction, increasing the cohesion and survival probability of groups. As chemical communication evolved, it complemented and enhanced other sensory systems. For example, the integration of chemical signals with visual input from emerging eyes allowed animals to form a more comprehensive picture of their environment. This multisensory approach led to nuanced behaviors, such as choosing habitats based on chemical and visual information that enhanced survival and ecological specialization. In neural terms, chemical detection involves converting chemical stimuli into electrical signals transmitted to nerves and processed by the brain or simpler ganglia. The refinement of this signaling pathway paralleled the development of nervous systems, representing a critical intersection of sensory biology and behavior. Fossil evidence for chemical communication is indirect but compelling. Trace fossils showing coordinated movements, evidence of spawning beds, and comparisons with living relatives provide clues to chemical signaling's role. For example, modern marine invertebrates often use chemical cues extensively, suggesting that their ancient ancestors did likewise. This early chemical communication set vital ecological and evolutionary precedents. It fostered complex interactions, predator alertness, social aggregation, mating systems that continue to define animal life. Chemical sensing was a silent architect, shaping habitats, behaviors, and evolutionary paths beneath the waves. As we ponder this elusive chemical language, we recognize it as a primordial form of awareness, a vital thread connecting life's earliest moments to the intricate sensory worlds of today. Next, we will explore a remarkable chapter in the Ordovician seas, where this sensory sophistication coincided with an extraordinary evolutionary development: the emergence of the first shelled cephalopods and the advent of jet propulsion, innovations that revolutionized mobility and predation strategies far beyond the Cambrian horizon. As the Cambrian seas gave way to the Ordovician period, approximately 485 to 444 million years ago, marine life underwent an impressive expansion and diversification, setting the stage for some of the most remarkable evolutionary innovations in the ancient oceans. Among these developments was the emergence of the first shelled cephalopods, a group of mollusks whose descendants include modern squids, octopuses, and nautiluses. This epoch effectively witnessed the dawn of sophisticated aquatic propulsion systems and advanced predatory adaptations that revolutionized marine ecosystems far beyond the achievements of earlier periods. The earliest shelled cephalopods descended from simple, conical ancestors, likely resembling straight-shelled nautiloids. Unlike many predecessors, these creatures developed external, chambered shells that provided buoyancy control and protection from predators. Their shells were composed primarily of aragonite, a form of calcium carbonate, with multiple partitions called septa dividing the shell into compartments. By regulating gas and fluid within these chambers, cephalopods could adjust their buoyancy precisely, allowing them to ascend, descend, or hover in the water column with remarkable ease. This ability to control buoyancy was the foundation for an unprecedented mastery over three-dimensional movement. Instead of dwelling near the sea bottom, like many Cambrian ancestors, shelled cephalopods took to the open waters, occupying pelagic zones where mobility and speed became vital for survival, both as predators and prey. Crucial to their swimming prowess was the development of jet propulsion, a highly efficient locomotive method unique among invertebrates. Jet propulsion operates on the principle of expelling water forcefully through a siphon-like funnel, generating reactive thrust that propels the animal backward rapidly. The muscular mantle surrounding their body contracted rhythmically, drawing water into a cavity and then expelling it jet-like. allowing for bursts of swift movement. This mode of propulsion offered tremendous advantages. It enabled quick acceleration to escape predators or to chase down prey. The ability to maneuver with agility in three-dimensional space was a game-changer, enabling cephalopods to exploit niches that less mobile animals could not, actively pursuing elusive animals or darting away from threats in complex seascapes. Anatomically, Ordovician cephalopods retained some primitive features, but also began evolving characteristics associated with heightened predatory efficiency. Their heads housed well-developed tentacles, used for grasping prey and manipulating objects. Though the full evolution of complex eyes and nervous systems would take longer, early cephalopods already exhibited improved sensory capacities that complemented their mobility. Fossil evidence from this time, including graceful, straight-shelled orthocarids and others with gently curved shells, reveals a diverse array of ecological strategies. Some species were probably ambush predators, using stealth and sudden bursts of speed to capture prey, while others might have scavenged or foraged between hunting bouts. These early cephalopods played a vital role in Ordovician food webs, introducing new levels of predation intensity that cascaded through marine communities. Their mobility and hunting prowess pressured other animals to further enhance defensive features, promoting evolutionary innovations across taxa. Interestingly, the Ordovician expansion coincided with broader environmental changes, rising sea levels, increased continental shelf areas, and temperate climates that created vast new marine habitats. Shelled cephalopods took full advantage of these opportunities, rapidly diversifying and spreading globally. Their success also laid the groundwork for future cephalopod evolution. Over millions of years, descendants would lose external shells, gaining even greater mobility and predatory skills, ultimately becoming some of the ocean's most formidable hunters. Visualize the Ordovician Ocean as a dynamic theater, cephalopods jetting gracefully through expansive waters, their shimmering shells glinting in diffuse sunlight, pursuing prey with newfound intent and agility. This scene embodies a thrilling evolutionary leap, a fusion of biological innovation and environmental opportunity that propelled marine life into uncharted realms. As we reflect on this transformative era, we prepare to next explore the reign of the giant sea scorpions, the Eurypterids, whose aquatic dominance would echo these themes of adaptation and power. But before then, we appreciate how the Ordovician shelled cephalopods harnessed the forces of nature to become masters of movement, signaling the continual march of life's ingenuity. As the Ordovician period unfolded and marine ecosystems flourished with new inhabitants, a group of formidable predators rose to dominance in the ancient seas: the giant sea scorpions, or Eurypterids. These remarkable arthropods, related to modern scorpions and spiders, carved a niche for themselves as some of the most powerful and influential marine creatures before the age of dinosaurs, reigning supreme throughout the Paleozoic era. Their story is one of evolutionary innovation, fierce predation, and aquatic mastery that left an indelible mark on prehistoric oceans. Eurypterids first appeared in the early Ordovician, about 460 million years ago, and thrived for over 250 million years until their decline near the close of the Permian. Their fossil record reveals a wide range of sizes and forms, but some species achieved staggering dimensions, with the largest, such as Ecological, reaching lengths of over 2.5 meters, approximately 8 feet. In a world where many animals remained small or moderate in size, these giants were true behemoths preying on a variety of marine life. Anatomically, Eurypterids possessed streamlined armored bodies segmented into a prosoma, head, an opisthosoma, abdomen, and a distinctive, often spiked telson, the tail spine. Their exoskeletons provided vital protection, much like the armor of trilobites. but they were combined with flexible joints enabling impressive agility. Adapted for aquatic life, many eurypterids had paddle-like appendages specialized for swimming, allowing swift and controlled movement through the water. Their sensory systems were equally evolved. Compound eyes equipped with numerous lenses offered a panoramic field of vision, enabling eurypterids to detect prey, predators, and mates with remarkable acuity, even in diverse light conditions. Alongside visual cues, their exoskeleton bore sensory hairs attuned to vibrations, enhancing their ability to interpret the surrounding waters. One of the most extraordinary aspects of eurypterids was their varied and effective predatory arsenal. Their large, claw-like appendages were adapted for grasping, crushing, or slicing prey. Some species wielded powerful pincers to seize soft-bodied animals, while others had fine, comb-like limbs useful for filtering food or stirring sediment. Their feeding strategies were diverse, ranging from active hunters pursuing agile prey to ambush predators lying in wait among reefs and seafloor structures. Paleontological evidence, including fossilized gut contents, illustrates that eurypterids preyed upon a broad spectrum of animals, from trilobites and early fish to smaller eurypterids. Their ecological role was that of apex predators, shaping prey populations and influencing community structures. The presence of these giants likely spurred further evolutionary adaptations among contemporaneous animals, including enhanced defense mechanisms and escape behaviors. Eurypterids also exhibited behavioral adaptations that enhanced their dominance. Some species are suspected to have ventured into brackish or even freshwater environments, showcasing physiological versatility rare among their marine peers. Such ecological flexibility might have provided refuge from competition and opened new hunting grounds. Reproductive behaviors, although not extensively documented, can be inferred from trace fossils suggesting complex mating rituals and nursery areas. Some eurypterids possibly displayed sexual dimorphism, with males possessing larger pincers or differently shaped appendages, hinting at intricate social interactions beyond mere survival. The extinction of eurypterids towards the end of the Permian likely stemmed from massive environmental upheavals during the largest mass extinction event in Earth's history. Their decline marked the end of an era, but paved the way for other marine predators to rise, including early sharks and later marine reptiles. Imagining the ancient seas where eurypterids ruled, one can picture vast armored beasts gliding powerfully through the water, their eyes scanning the depths for prey, pincers poised for the decisive strike. Their legacy is a testament to the power of adaptation and the dynamic nature of ecological dominance in the prehistoric world. Their story connects us to the broader tapestry of life's history, exemplifying how innovation in form, function, and behavior allows species to thrive and shape their environments. As we proceed, we will explore the fascinating architects of the first reefs, whose community building and structural innovations added yet another dimension to marine ecosystems, setting the stage for life's increasing complexity beneath and upon the waves. As we emerge from the reign of powerful predators like eurypterids, our exploration shifts to another cornerstone of ancient marine ecosystems: the first reefs. These remarkable structures, built by coral-like organisms and other early architects, fostered vibrant community living beneath the waves and transformed the seafloor into dynamic, complex habitats. The advent of reef building represents not merely a geological phenomenon, but a profound biological innovation that shaped ecological interactions and biodiversity for millions of years. Unlike solitary organisms drifting or crawling through the seas, reef builders created substantial, stable, and intricately structured frameworks by secreting hard exoskeletons or skeletons over generations. These physical edifices provided shelter, breeding grounds, and feeding opportunities for a multitude of species, effectively serving as biodiversity hotspots. The origins of reef construction trace back to the late Cambrian and flourished through the Ordovician and beyond. Early reef-like structures were composed predominantly of calcareous sponges, stromatoporoids, and certain calcifying algae. Most notably, primitive coral-like animals, early relatives of today's corals, began secreting calcium carbonate to form protective external skeletons, facilitating the growth of extensive reef habitats. One of the pioneering architects was the stromatoporoid, a sponge-like organism characterized by layered skeletal structures that anchored the reef and provided robust frameworks. These creatures, alongside calcifying algae, stabilized sediments and permitted three-dimensional reef growth, elevating sections of the seafloor into complex topographies. Such structures increased habitat heterogeneity, inviting colonization by a growing variety of animals. The burgeoning reefs not only offered refuge from predators, but also concentrated food resources by filtering plankton and organic particles. This concentration created ecological niches that amplified species interactions, fostering symbiosis, competition, and cooperation within these underwater cities. Crucially, reef ecosystems nurtured early forms of community living. Organisms inhabiting reefs often formed colonies or aggregated communities, benefiting from proximity to conspecifics and unrelated species alike. These assemblages enabled the exchange of resources and information, potentially leading to primitive social behaviors and cooperative strategies that enhanced survival. The complexity of the first reefs even influenced evolutionary trajectories. Species adapted to life within or around reefs developed specialized morphologies, such as flattened bodies to slip between crevices or spiny exteriors to deter predators lurking in confined spaces. Moreover, the challenge of growing in 3 dimensions spurred innovations in growth patterns and reproductive strategies. Importantly, reef ecosystems played a pivotal role in biogeochemical cycles. Through the accumulation of calcium carbonate, reefs contributed to carbon sequestration, influencing ocean chemistry and long-term climate regulation. This environmental engineering underscores the extensive impact of biological activity on Earth's systems. Fossilized reefs from sites like the Ordovician reefs in North America and ancient coral fossil beds worldwide offer vivid glimpses into these thriving ecosystems. These geological archives capture the intricate interplay of architecture, biology, and environment characteristic of early reef communities. Visualize the ancient seafloor transformed by towering mounds and labyrinthine caves, habitats teeming with diverse animal life, ranging from filter-feeding sponges and mollusks to mobile predators and scavengers. This underwater metropolis hums with activity, interactions, and evolutionary opportunity, a foundation upon which modern coral reefs and marine ecosystems are built. As the narrative of life's early marvels continues, we next explore innovators with shells, the evolution of protective and mobile encasements that allowed animals to thrive in a wide range of habitats. These developments, intimately linked with reef ecosystems and predation pressures, reveal the intricate dance between offense and defense that drives natural history forward. In the wake of thriving reef ecosystems and the burgeoning complexity of marine life, another evolutionary marvel unfolded with profound consequences: the emergence and diversification of animals adorned with shells. These innovators with shells not only enhanced their defense against predation, but also revolutionized mobility and ecological roles in ancient waters. The evolutionary journey of protective exoskeletons or shells is one of adaptation, resilience, and the delicate balance between safeguarding life and maintaining the agility necessary for survival. The Cambrian Explosion and subsequent Ordovician expansions set the evolutionary stage where shelter and survival became inseparable. As predation pressures intensified and environments grew more diverse, animals faced a distinct survival challenge: how to defend themselves without sacrificing the ability to move, feed, or reproduce efficiently. Shells, mineralized outer coverings, emerged as elegant solutions to this challenge. Early shells were primarily composed of calcium carbonate and, in some cases, silica, providing a rigid barrier against physical assaults. These coverings ranged from simple, single-piece plates to more intricate, multi-chambered structures. The presence of such shells wasn't limited to a single group. Mollusks, brachiopods, and various arthropods independently evolved these protective armors, a testament to the convergent evolutionary importance of this trait. Mollusks, for example, displayed a remarkable evolutionary versatility in their shells, from the humble limpets adhering to rocks with simple conical shells to the more elaborate cephalopod shells that allowed buoyancy control and advanced mobility. Brachiopods developed bilateral shells enclosing their soft bodies, not unlike modern clams, but distinct in anatomical structure. Arthropods like trilobites had calcified exoskeletons segmented for flexibility and protection. These shells shielded vulnerable body parts while enabling movement thanks to articulated joints or overlapping plates. The relationship between protection and mobility reached a new zenith with adaptations that balanced these competing needs. Some animals evolved shells with streamlined shapes to reduce water resistance, facilitating faster or more energy-efficient swimming. Others developed specialized joints or hinge mechanisms to allow shell opening and closing, providing both defensive closure and an ability to interact with their environment. Moreover, the evolution of coiled shells in some mollusks like the gastropods introduced an innovative way to compactly protect the organism while retaining center of gravity and balance during movement. This shape also offered mechanical advantages when withdrawing from harm. Shells also played a significant role beyond mere defense. They served as platforms for muscle attachment, enabling stronger and more precise movements, whether crawling, burrowing, or swimming. The hardened exterior gave organisms leverage, enhancing locomotive capacity and allowing exploration of various habitats, including soft sediments, open waters, and reef crevices. Evolutionary pressure from predation spurred further refinement of shells. Spines, ridges, serrations, and ornamentations evolved on shells to deter predators physically or through visual intimidation. Some shells developed thickness variations strategically distributed to protect vital parts, while maintaining reduced weight in less critical areas to support agility. The appearance of mobile, shell-bearing animals prompted a cascade of ecological shifts. Herbivores began exploiting previously untouched niches, and predators evolved strategies to bypass or penetrate hard defenses, fueling an ongoing evolutionary arms race. This dynamic interplay molded trophic structures and community complexity in ancient marine ecosystems. Fossils reveal the diversity and durability of these evolutionary innovations, showcasing creatures that thrived in their armored repast. Their shells even created lasting geological signatures, contributing calcium carbonate to sediment layers, influencing sedimentology and paleoceanography. Envision the ancient seas, alive with armored voyagers gliding, crawling, and burrowing, each shell a fortress, a tool, and a key to survival in a world of relentless challenges. Their coexistence reflects the exquisite balancing act of evolution, where protection and mobility intertwined to sculpt life's myriad forms. As we reflect on these early innovators with shells, our journey soon leads us to a monumental chapter: the colonization of land by ancient arthropods during the Silurian. This pivotal move onto dry terrain leveraged many of the adaptations forged in aquatic realms, marking yet another triumphant expansion of life's frontiers. The journey from aquatic realms to the terrestrial world represents one of the most monumental shifts in the history of life on Earth. Among the pioneers of this bold venture were ancient arthropods, creatures whose evolutionary innovations enabled them to transcend the boundaries of water and embark on a Silurian safari across the nascent land masses that emerged roughly 443 to 419 million years ago. This remarkable transition not only transformed the landscapes they traversed, but also laid the foundational footsteps for future terrestrial life. The Silurian period followed the dramatic Ordovician biodiversification, which expanded marine ecosystems and set complex ecological dynamics into motion. But it was in the relatively stable and warm climatic conditions of the Silurian that the stage was set for life to conquer land. Early terrestrial habitats were starkly different from marine environments, exposed to desiccation, temperature fluctuations, increased ultraviolet radiation, and the absence of buoyancy. Overcoming these challenges required a suite of anatomical, physiological, and behavioral adaptations, many of which the resilient arthropods began to develop and refine. Arthropods, a phylum characterized by jointed appendages, segmented bodies, and exoskeletons, were pre-adapted in many ways for terrestrial life. Their segmented exoskeletons provided mechanical support against gravity and a barrier to water loss, critical points in the harsh, dry terrestrial environment. The exoskeleton's waxy epicuticle limited desiccation, while their articulated limbs allowed effective terrestrial locomotion over uneven substrates, from moist soils to rocky outcrops. Among the earliest colonizers were myriapods, the ancestors of centipedes and millipedes, arachnids, including spiders and scorpion-like creatures, and hexapod-like organisms that would eventually give rise to insects. Fossil and trace evidence suggests these arthropods ventured into moist ecosystems near water bodies, such as riverbanks, deltas, and wetlands, where humidity buffered the risks of dehydration. One of the key physiological hurdles was respiration. While aquatic ancestors used gills to extract oxygen dissolved in water, terrestrial arthropods evolved respiratory structures adapted to air. Early arachnids developed book lungs, which are layered respiratory organs capable of gas exchange in air, balancing oxygen uptake with water conservation. Myriapods and later insects evolved tracheal systems, intricate networks of tubes delivering oxygen directly to tissues, minimizing water loss and allowing higher metabolic rates suitable for terrestrial activity. Sensory adaptations were equally crucial. For navigation and survival on land, early arthropods refined mechanoreceptors that detected vibrations transmitted through solid substrates and chemoreceptors attuned to airborne chemical cues. This sensory evolution allowed them to find food, avoid predators, and communicate in the three-dimensional terrestrial environment. Behaviorally, early terrestrial arthropods exploited niche habitats where competition was low and resources ample. Detritivores like primitive millipedes fed on decaying plant material, contributing to nutrient cycling and soil formation. Predatory groups such as centipede ancestors took advantage of abundant small invertebrates. This ecological diversification mirrored marine food webs, but adapted to the land's unique constraints and opportunities. Fossil trackways dating back to the Silurian, such as those found in recent discoveries from ancient tidal flats and river margins, provide compelling evidence of terrestrial arthropod mobility. These trace fossils capture footprints and dragging marks, revealing patterns of locomotion and interactions with the environment. They paint vivid pictures of early land exploration, a world bustling with the first arthropod forays beyond water's edge. The colonization of land by arthropods also had cascading effects on terrestrial ecosystems. Their burrowing and feeding activities aided soil aeration and organic matter breakdown, facilitating the establishment of the earliest terrestrial plants. In turn, these plants stabilized soils and created microhabitats, supporting further expansion and diversification of terrestrial fauna. This Silurian safari was not merely a gradual creep onto land, but a transformative leap, where the evolutionary toolbox of arthropods was adapted and augmented to meet unprecedented environmental challenges. The success of this conquest foreshadowed the later dominance of insects and spiders, groups whose ecological and evolutionary breadth has shaped terrestrial life up to the present day. Imagine then the silent march of ancient arthropods across verdant, primitive land, a pioneering safari under the Silurian sun, bodies armored against dehydration, limbs delicately treading upon the soil, sensory organs capturing the scents and vibrations of a world coming alive. These first explorers bridged the aquatic and the terrestrial, unraveling new ecological potentials and setting in motion the wondrous story of life's terrestrial odyssey. As we consider these groundbreakers, our next exploration will delve into the vital anatomical innovations that complemented terrestrial living: the advent of breathing air through tracheal systems and lungs. These respiratory breakthroughs empowered life to thrive on land and heralded new chapters of evolutionary possibility and complexity. As terrestrial life forged ahead on the solid grounds of the Silurian and Devonian periods, a monumental biological innovation facilitated the profound transformation of air breathing: the advent of tracheal systems and lungs. This evolutionary leap not only liberated animals from their ancestral aquatic constraints, but also opened vast new ecological opportunities on land. Understanding how respiratory structures evolved offers a window into the intimate relationship between physiology, environment, and the relentless drive of life to conquer new frontiers. Respiration, the ability to absorb oxygen and expel carbon dioxide, is fundamental to energy production in all complex animals. In aquatic environments, this exchange commonly occurs through gills, delicate organs optimized to extract dissolved oxygen from water. However, water and air differ substantially in their oxygen content, moisture levels, and physical properties. Transitioning from water to air required revolutionary adaptations to cope with desiccation and effectively capture oxygen in a gaseous milieu. The earliest terrestrial arthropods, descendants of aquatic ancestors, began by modifying existing structures to accommodate air breathing. The insects and myriapods' ancestors evolved tracheal systems, networks of minute tubes known as tracheae, that permeated the body and delivered oxygen directly to cells. Air entered these tubes through external openings called spiracles, which could open or close to reduce water loss, a decisive adaptation in dry environments. Tracheal systems represented a breakthrough in respiratory efficiency, particularly for small-bodied organisms. By bypassing the limitations of circulatory transport to diffuse oxygen directly to tissues, these animals attained higher metabolic rates suitable for sustained activity on land. This respiratory innovation became the foundation for the immense diversification and ecological success of insects, the most numerous and varied group of terrestrial animals. Parallel to the evolution of tracheae, another respiratory adaptation emerged primarily in early vertebrates: lungs. Lungs originated as outpocketings of the gut, rudimentary air sacs that extracted oxygen from the atmosphere while minimizing moisture loss. Early lobe-finned fishes, a group ancestral to tetrapods, developed primitive lungs as supplements to gills, allowing survival in oxygen-poor water and periodic excursions to the surface for air gulping. The Devonian period, often dubbed the Age of Fishes, witnessed the diversification of these lung-bearing fishes. Some genera, like Tiktaalik and Eusthenopteron, exhibit features indicative of dual respiration, with both gills and rudimentary lungs. This physiological flexibility was vital for venturing into shallower, often hypoxic waters, providing a stepping stone toward permanent terrestriality. As vertebrates gradually took their first steps onto land, lungs became increasingly specialized and elaborate. Enhanced vascularization, greater surface area, and structural compartmentalization improved gas exchange efficiency, supporting larger body sizes and more complex metabolisms. The development of a muscular diaphragm in later tetrapods further refined the mechanics of breathing, allowing active ventilation rather than passive diffusion. The ramifications of evolving tracheal systems and lungs extended beyond simple oxygen uptake. These respiratory structures influenced circulatory system development, metabolic capacity, and even behavioral patterns. Animals could sustain longer forays away from water, explore diverse habitats, and engage in activities requiring bursts of intense energy, hunting, mating displays, or territorial defense. Environmental challenges like fluctuating humidity, temperature, and atmospheric oxygen concentration spurred further respiratory innovations. Spherical regulation evolved to balance oxygen supply with water conservation. Lungs adapted to prevent collapse and maintain moisture. Some arthropods developed waxy body coverings complemented by respiratory refinement to minimize dehydration. The emergence of air-breathing systems also reshaped ecological communities. Plants colonizing land created new oxygen dynamics through photosynthesis and altered soil conditions. Animals with tracheal systems or lungs participated in novel symbiotic relationships, nutrient cycles, and food webs anchored on terrestrial productivity. Visualize early terrestrial landscapes with air-breathing pioneers, ancient insects navigating leaf litter with intricate tracheal networks sustaining flight muscles, early amphibians gulping air, lungs expanding rhythmically as they explored shallow pools and riverbanks. These first breaths on land symbolize a pivotal chapter in Earth's biologic narrative, the mastering of a wholly new environment. As we deepen our appreciation for respiratory evolution, we prepare to explore the rise of early herbivores, the birth of plant-eating strategies that would endlessly intertwine animal and plant life, accelerating terrestrial ecosystem complexity. This mutual evolution set the stage for diverse habitats, trophic dynamics, and ultimately the flourishing of terrestrial biodiversity that defines our world today. The arrival of plants on land marked one of the most transformative events in Earth's natural history, reshaping barren landscapes into verdant habitats teeming with life. Yet, equally revolutionary was the emergence of the animals that began to feed upon these plants, the early herbivores. This pivotal development, spanning the Silurian and Devonian periods, heralded new ecological interactions and evolutionary pressures that would echo throughout terrestrial ecosystems for millions of years. Before animals ventured onto land, plants had already begun their terrestrial conquest. Simple, non-vascular plants like mosses and liverworts appeared first, gradually giving way to more complex vascular plants, including the earliest primitive trees and shrubs by the Devonian. These plants transformed soil quality, stabilized erosion, produced oxygen, and created habitats that beckoned life's colonizers. With abundant vegetation came the opportunity and the challenge for herbivory to evolve. Feeding on plants requires distinct adaptations because plant material is structurally tough, often chemically defended, and nutritionally different from animal prey. Early terrestrial animals had to overcome physical barriers like cellulose and lignin, as well as chemical deterrents such as toxins or bitter compounds. Among the earliest herbivores were certain arthropods, especially primitive myriapods, and rhiniognaathids, ancient relatives of modern insects. Fossil evidence suggests that some of these creatures adapted mouthparts capable of chewing or scraping plant surfaces. Hardened mandibles, piercing jaws, and specialized grinding plates evolved to process fibrous plant tissues. This marked the birth of specialized plant-eating strategies, distinct from predatory or scavenging feeding. Behavior also played a role in herbivory's early evolution. Some animals grazed on low-lying mosses and ground cover, while others might have climbed primitive plants to feed on higher leaves and stems, encouraging further plant-animal interactions. These feeding behaviors could influence plant morphology, selecting for traits such as tougher leaves, thorns, or rapid regrowth, sparking early coevolutionary arms races between flora and fauna. The advent of herbivory dramatically impacted nutrient cycles. By consuming plants, animals facilitated the breakdown and recycling of organic matter. Their waste products enriched soils and supported microbial communities, promoting ecosystem productivity and complexity. Herbivory also opened novel ecological niches and fostered diversification. Plants and their consumers formed intricate food webs where plant availability could dictate herbivore populations, which in turn supported higher-level predators. Fossilized gut contents and trace fossils bearing feeding marks provide crucial insights into these ancient diets. Such evidence reveals bite patterns, plant fragment delivery, and digestive adaptations unique to herbivores. It is important to appreciate that early herbivory was likely opportunistic and modest in scale, gradually intensifying as both animals and plants evolved in complexity. These pioneering plant eaters set in motion ecological dynamics that would culminate in vast forests teeming with insects, reptiles, mammals, and other consumers. Picture a Devonian landscape where armored, multi-legged arthropods cautiously nibble at the first verdant stalks, molding the ecosystem around them. Their emergence signifies nature's versatile creativity, turning simple plants and primitive animals into a complex dance of energy flow and life-sustaining interdependence. Our next step in this evolutionary saga explores the advent of jawed fishes, the placoderms, and their importance as armored pioneers in aquatic ecosystems. These fishes introduced new predatory strategies and robust body plans that were crucial for the further evolution of vertebrate life. As life continued its profound evolutionary journey in the Devonian period, often heralded as the Age of Fishes, one group stood at the forefront of a pivotal transition: the placoderms. These armored pioneers were among the first jawed fishes, marking a fundamental shift in vertebrate evolution that would echo through subsequent eras, eventually paving the way for the emergence of sharks, bony fishes, and ultimately, terrestrial vertebrates. Placoderms were characterized by their distinctive armor plating, bony shields covering the head and thorax regions, providing formidable protection against predators. Unlike earlier jawless fishes, these creatures developed hinged jaws, enabling them to bite, grasp, and process a wider variety of food. This innovation redefined feeding strategies in aquatic ecosystems, allowing placoderms to exploit new prey types and ecological niches. The jaw's origin is an evolutionary marvel. It likely evolved from the modification of the first pair of gill arches in jawless ancestors. This anatomical adaptation not only improved feeding efficiency, but also facilitated aggression and defense, influencing the balance of power in ancient aquatic communities. Aside from their armored heads, placoderms exhibited a range of body forms. Some were relatively slow, benthic dwellers feeding on detritus and small invertebrates, while others, such as the famed Dunkleosteus, grew to gigantic sizes, some exceeding 6 meters in length, and were apex predators capable of delivering bone-crushing bites. Dunkleosteus, perhaps the most iconic placoderm, earned its reputation for both massive armored plates and extraordinarily strong jaw muscles. Fossil evidence suggests its bite force surpassed many modern-day predators, enabling it to prey upon almost any contemporaneous marine animal, including other armored fishes. Placoderms didn't just influence the food web through their predatory roles, they also displayed reproductive innovations rare for their time. Fossils reveal evidence of internal fertilization and live birth in some species, illustrating complex life history traits. The rise and diversification of placoderms coincided with rich marine habitats and reef systems that provided opportunities and challenges. Their armor and jaws allowed them to dominate certain niches, driving evolutionary arms races that elevated predator and prey adaptations alike. However, despite their success, placoderms eventually declined and went extinct by the end of the Devonian, likely due to environmental shifts and competition with emerging jawed fishes lacking heavy armor but possessing other advantages. Their legacy lies not only in their fossilized remains, but also in their profound influence on vertebrate evolution. By introducing jaws and formidable protective armor, placoderms set the stage for the complex marine ecosystems that would follow. They represent a crucial evolutionary bridge between primitive jawless vertebrates and the diverse jawed lineages dominating seas and rivers today. Imagine the Devonian seas teeming with armored giants and agile hunters, where the evolution of jaws changed the very dynamics of survival and feeding. This era surges with energy and adaptation, setting the evolutionary foundation for more complex life. Our narrative now advances to a fascinating exploration of ancient toxicity and venom systems, uncovering how early animals harnessed biochemical weapons for predation and defense, revealing yet another dimension of the evolutionary arms race preceding the age of dinosaurs. As the tapestry of ancient life continued to evolve in complexity and interconnection, one of the most fascinating and effective innovations to arise was the development of venom and toxicity as methods of predation and defense. These biochemical weapons emerged early in animal evolution, providing species with specialized tools to capture prey, deter predators, and compete within increasingly dynamic ecosystems. Understanding the origins and roles of venoms and toxins among prehistoric animals sheds light on the ingenious ways life adapted to survive and thrive long before the reign of the dinosaurs. Venom systems represent a remarkable convergence of biology and chemistry, where organisms produce complex mixtures of proteins, peptides, and enzymes designed to disrupt physiological functions of other creatures. The evolutionary pressures driving the emergence of venom were immense. Predators needed efficient means to immobilize or kill prey swiftly, while prey animals sought ways to discourage or incapacitate attackers. These chemical arsenals could accomplish these ends with precision and often minimal physical confrontation, conferring distinct survival advantages. Among the earliest known venomous animals were marine invertebrates such as cnidarians, jellyfish, sea anemones, and corals, which utilize specialized cells called cnidocytes. These cells contain harpoon-like nematocysts loaded with venomous compounds that instantly immobilize or deter other animals upon contact. Remarkably, fossils and molecular studies suggest that these venom systems date back more than 500 million years, placing them at or even before the Cambrian Explosion. As predation intensified during the Cambrian and Ordovician, venoms became increasingly sophisticated and widespread. Some arthropods, ancestors of modern spiders and scorpions, evolved venom glands associated with pincers or stingers, allowing them to subdue prey immobilized by chemical paralysis. These adaptations were critical in terrestrial and aquatic hunting success, especially given the limitations on size and strength. In the aquatic realm, certain early fish groups also developed toxins. For example, some placoderms, the armored fishes of the Devonian, exhibited bony spines and muzzle structures that might have harbored venomous secretions, though definitive evidence is sparse and an exciting frontier for paleontological research. Venomous strategies diversified further with the appearance of complex delivery mechanisms, fangs in early gnathostoms, jawed vertebrates, stingers in arthropods, and specialized spines or barbs capable of injecting toxins deep into tissues. These biological syringes enhanced effectiveness, allowing precise targeting of foes or prey. Beyond direct predation, toxicity served as a powerful defense. Bright coloration and distinctive patterns evolved in some toxic species, creating aposematic signals that warned potential predators of danger. This visual communication reduced unnecessary conflicts and conserved energy for all parties. Chemical mimicry arose as well, where non-toxic animals evolved appearances or behaviors resembling venomous ones to gain protective benefits, weaving a complex ecological web based on chemical signaling. Importantly, venom and toxins are not static. They evolve rapidly due to ongoing evolutionary arms races. Prey species can develop resistance or behavioral avoidance, prompting predators and venomous animals to refine their chemical cocktails. This dynamic interplay has driven tremendous diversification in venom composition and function over hundreds of millions of years. Modern molecular biology has revealed that many venom components derive from genes recruited and repurposed from ordinary physiological proteins, emphasizing evolution's resourcefulness in innovation. Envision primordial seas and early terrestrial landscapes peppered with creatures wielding biochemical weapons. Jellyfish, lashing stinging tentacles, scorpions poised to deliver venomous stings, and predatory fishes with venomous spines. These strategies added a subtle but potent layer to survival tactics, influencing the behavior and evolution of myriad species. As we contemplate these ancient chemical arsenals, we prepare to examine the remarkable diversification during the Devonian period, where limbs and early tetrapods appeared, heralding the nascent conquest of land by vertebrates and setting the stage for terrestrial vertebrate evolution that would culminate in the age of dinosaurs. The Devonian period, often called the Age of Fishes, was a dynamic epoch that witnessed not only an explosion of fish diversity, but also a monumental transition that bridged aquatic and terrestrial life, the evolution of limbs, and the origin of the earliest tetrapods. This evolutionary leap transformed vertebrate anatomy and ecology, setting the stage for vertebrates to eventually conquer land and establish a dominant terrestrial presence. The Devonian seas and rivers teemed with an extraordinary variety of fishes exhibiting an array of body plans and ecological strategies. Among these were the lobe-finned fishes, or sarcopterygians, whose fleshy, lobed fins with robust internal bones represented a crucial anatomical innovation. Unlike the ray-finned fishes, whose fins consist primarily of thin rays supportive of delicate membranes, lobe-finned fishes had muscular, limb-like fins capable of bearing weight and enabling complex movements. Fossil representatives like Eusthenopteron and Panderichthys showcase these transitional features remarkably well. Their fins contained bones homologous to the humerus, radius, and ulna of later limbs, illustrating the gradual modification from fin to limb. These fishes likely inhabited shallow, vegetated waters, where robust lobed fins could facilitate movement through dense aquatic plants or across muddy substrates, a precursor to terrestrial locomotion. As these fishes evolved, their fins gained functional versatility. They could not only propel the animal through water, but also enable it to prop itself up or maneuver in environments where swimming alone was insufficient. This adaptability was critical in dynamic, oxygen-poor Devonian wetlands and waterways, which often required breath-holding and movement in confined spaces. The gradual transformation culminated in the evolution of early tetrapods, vertebrates with 4 limbs equipped with digits, providing enhanced stability and mobility on land. Ichthyostega and Acanthostega are iconic Devonian tetrapods exhibiting a blend of aquatic and terrestrial adaptations, limbs with digits capable of supporting weight, lungs for air breathing, but retaining gills and aquatic life habits. This phase reflects a pivotal evolutionary compromise. Early tetrapods remained tied to water for reproduction and certain life functions, yet their limbs and lungs allowed explorations of terrestrial environments such as floodplains, shallow ponds, and marshes. This ecological flexibility gave them a considerable advantage in exploiting new niches and avoiding aquatic predators or competition. Anatomical innovations accompanying limbs evolution included strengthened vertebral columns, modified shoulder and pelvic girdles to support body weight outside water, and changes in muscle arrangement favoring terrestrial locomotion. These adaptations reflect an intricate remodeling of the vertebrate body plan to meet terrestrial mechanical demands. Importantly, the Devonian diversification of limbs and early tetrapods was not a single linear progression, but a complex radiation, with multiple lineages experimenting with different degrees of terrestriality and limb structures. Such diversity illustrates that the water-to-land transition was a mosaic of adaptations responding to varied ecological pressures. The proliferation of limbs also partnered with sensory and respiratory innovations, enhancing environmental awareness and survival capabilities on land. Early tetrapods likely developed enhanced vision in air, more sophisticated hearing, and improved olfaction, facilitating nuanced interactions within their novel habitats. The Devonian limb evolution set a precedent that would radiate through vertebrate evolution, ultimately giving rise to amphibians, reptiles, birds, and mammals. It symbolized one of evolution's most profound explorations of form and function, embodying life's tenacity and adaptability. Picture the twilight of the Devonian landscape. Shallow rivers meander through lush floodplains, their banks alive with early tetrapods cautiously venturing onto land, limbs pressing against muddy substrates, lungs drawing in fresh air. This scene captures a transformative moment, a hinge between worlds that heralded life's terrestrial ascendancy. Looking ahead, our journey will next illuminate the first steps on land, chronicling how these pioneering tetrapods further adapted from their watery origins to become wholly terrestrial, marking a new epoch in the saga of life before the dinosaurs. Following the remarkable evolutionary developments of the Devonian, we arrive at a pivotal chapter in the history of life on Earth. The first steps taken by tetrapods as they transitioned from water to land. This monumental journey did not happen overnight, but through a gradual series of adaptations, modifications, and ecological experiments that reshaped vertebrate anatomy and behavior. It opened entirely new frontiers for life, laying the groundwork for the terrestrial ecosystems we recognize today. Tetrapods, literally four-footed vertebrates, originated from lobe-finned fishes whose robust limb-like fins offered the structural foundation for limbs. These early tetrapods inhabited swampy, shallow aquatic environments that provided both oxygen challenges and opportunities to explore marginal habitats. The pressures and rewards of venturing onto land involved complex physiological, morphological, and ecological transitions. One of the central challenges was locomotion. Moving effectively on land required limbs capable of supporting body weight without the buoyancy of water. Early tetrapod limbs evolved stronger bones, with digits, fingers and toes, providing enhanced grip and balance. Fossils like those of Acanthostega and Ichthyostega reveal this balance. These animals retained aquatic traits such as gills and tail fins, yet their limbs had the capacity to propel them through mud and shallow water, and possibly even short forays onto land. They likely used their limbs to push through dense vegetation, navigate flooded terrains, or escape aquatic predators. Respiration marked another critical adaptation. Though lungs had evolved earlier as complements to gills, reliance on air breathing increased in these transitional vertebrates. This allowed them to survive in oxygen-poor water and eventually sustain time spent on land. The complex circulatory and respiratory changes accompanying this shift were vital for oxygen delivery under terrestrial conditions. Skin and moisture retention were important hurdles to overcome as well. Aquatic skin adapted to constant hydration was ill-suited for the drying air. Early tetrapods likely had thickened, glandular skin to reduce moisture loss, and behaviors such as seeking refuge in damp habitats to avoid desiccation. These traits would be further refined over millions of years. Sensory systems adapted to terrestrial environments exhibited remarkable specialization. Vision evolved to adjust to air's optical properties. Auditory systems began to process airborne sounds. Olfactory mechanisms tuned to detecting airborne chemicals. These sensory enhancements improved environmental awareness and predator-prey interactions on land. Reproductive strategies initially remained tied to water, with early tetrapods likely depositing eggs in aquatic or moist environments, limiting their full terrestrial independence. However, this constraint spurred innovations over geological time, including the development of amniotic eggs in later vertebrates, freeing them from reliance on water bodies for reproduction. From an ecological perspective, tetrapods transitioning to land opened new food sources and habitats. Early trophic relationships expanded with the inclusion of terrestrial invertebrates and plants. This ecological expansion fostered evolutionary radiation, with diverse vertebrate lineages emerging to exploit various niches. Imagine these pioneering tetrapods cautiously dragging themselves through marshy landscapes, limbs supporting their weight as they breathe fresh air and sense unfamiliar surroundings. Their movements symbolize a profound moment, the crossing of a threshold from aquatic life to terrestrial existence, a narrative of exploration, courage, and adaptation. This transition would ultimately seed the rich tapestry of terrestrial vertebrate evolution, from amphibians to reptiles, mammals, and birds. Next, we turn our attention to the evolution of supersenses out of water, exploring how hearing and smell developed and specialized to meet the unique challenges and opportunities of terrestrial life, further enhancing life's conquest of Earth's dry realms. As vertebrates and other animals ventured from aquatic habitats onto land, they encountered a world that was not only physically different, but also sensory-rich in new and challenging ways. The evolution of supersenses outside of water, particularly hearing and smell, became vital adaptations that enabled terrestrial animals to navigate, find food, detect predators, and communicate effectively in a vastly different environment. These sensory innovations ushered in a new era of interaction with the world around them, profoundly influencing animal behavior and ecology long before the rise of dinosaurs. In the aquatic world, sound propagation and chemical cues differ fundamentally from those in the air. Water is a denser medium where sound travels faster and carries well over long distances, but airborne sounds require different sensory structures to detect and process. Similarly, the detection of chemicals in air versus water involves alternative mechanisms due to differences in diffusion rates and volatility. Thus, as animals made the transition to terrestrial life, their sensory organs underwent remarkable transformations to meet these new environmental demands. Hearing, which had modest beginnings in aquatic ancestors as the ability to sense vibrations, became an increasingly acute and specialized sense on land. Early tetrapods likely evolved structures analogous to the middle ear, including the stapes bone, which initially functioned in transmitting vibrations from the jaw or skull, but was gradually co-opted to detect airborne sounds. This repurposing was crucial for perceiving the rich acoustic landscape of terrestrial habitats. Sounds of approaching predators, prey movement, or environmental noises such as wind through foliage. The evolution of the tympanic membrane, or eardrum, was another critical step in enhancing terrestrial hearing. This thin membrane vibrates in response to sound waves and relays these signals via ossicles to the inner ear, where hair cells transduce mechanical stimuli into neural signals. Fossil evidence and the study of extant amphibians illuminate the stepwise development of these auditory components, underscoring a narrative of gradual refinement tuned by selective pressures. Intimately linked to hearing was the anatomical and neurological evolution of the inner ear, including the cochlea, or its primitive counterparts. These structures not only filtered and enhanced sound signals, but also contributed to balance and spatial orientation, essential functions for coordinated movement on uneven terrestrial surfaces. Smell, or olfaction, similarly experienced an evolutionary renaissance upon the transition to land. While aquatic animals detect dissolved chemicals primarily via olfactory receptors, sensitive to waterborne molecules, terrestrial animals faced the challenge of perceiving volatile airborne compounds. To adapt, the nasal structures expanded and became more complex, with epithelial tissues specialized for volatile molecule detection. The development of nasal cavities and turbinates increased the surface area for odor detection and conditioned inhaled air by warming and humidifying it, key adaptations for survival in dry land environments. Olfaction became a primary modality for finding food, detecting mates through pheromones, and sensing predators or hazards. Many early terrestrial vertebrates exhibited enhanced smell capabilities, enabling them to thrive in nocturnal or densely vegetated conditions, where visual cues might be limited. In parallel, the integration of hearing and olfaction with other sensory inputs fostered complex behavioral responses and communication strategies. Early animals likely used acoustic signals and chemical cues not only for survival, but also for social interactions, territoriality, and reproduction. The evolutionary pressures shaping these supersenses were influenced by environmental heterogeneity. Dense forests, open plains, and subterranean habitats each imposed unique sensory challenges, prompting diversification in sensory organ morphology and function across different lineages. Contemporary comparative anatomy and genetics reveal conserved molecular pathways underlying sensory development, demonstrating deep evolutionary roots for hearing and smell that trace back to early vertebrates and invertebrate relatives. Imagine an ancient landscape where the first tetrapods cautiously interpret the rustle of leaves, the faint odor of prey on the wind, or the distant call of a potential rival. These sensory worlds, invisible yet vivid, framed the adaptive playground for early terrestrial ecological interactions. As sensory capabilities blossomed on land, the stage was set for ecological expansions and new life strategies. The next chapter in this evolutionary saga leads us into the Carboniferous period, a time marked by gigantism in insects and millipedes, showcasing how sensory adaptations paired with ecological opportunity to produce some of the most astonishing biological phenomena before dinosaurs dominated the Earth. The Carboniferous period, spanning roughly from 359 to 299 million years ago, stands as one of the most remarkable chapters in the history of terrestrial life, especially for the astounding gigantism exhibited by insects and millipedes. This era, characterized by vast swampy forests and high atmospheric oxygen levels, nurtured the evolution of some of the largest arthropods ever to walk or crawl the Earth. These giants were not only astonishing in size, but also exemplified how environmental conditions and physiological constraints combined to shape prehistoric life long before dinosaurs dominated the terrestrial realm. To understand gigantism in Carboniferous insects and millipedes, we first must appreciate the unique environmental context. The Carboniferous atmosphere contained oxygen concentrations surpassing 30%, significantly higher than the roughly 21% we experience today. This hyperoxic environment enhanced metabolic efficiency and oxygen diffusion, crucial for organisms like insects and millipedes that rely on passive diffusion through tracheal systems for respiration. The abundance of oxygen allowed them to grow larger without facing the oxygen limitations that constrain modern arthropod sizes. Among the most iconic Carboniferous giants was Arthropleura, a millipede-like arthropod that reached lengths exceeding 2 meters. Comprising some of the largest known land invertebrates in Earth's history, Arthropleura likely played a dominant role in the forest floor ecosystems, feeding primarily on abundant decaying plant matter and possibly engaging in some degree of herbivory. Its formidable size offered advantages, such as defense from predators and access to a wide range of food resources, alongside unique challenges, including mobility and structural support. Similarly impressive was Meganeura, a genus of giant dragonfly-like insects boasting wingspans surpassing 70 centimeters. These aerial predators patrolled the Carboniferous skies, preying upon smaller insects and filling ecological roles analogous to modern birds of prey or bats. The size of Meganeura and its kin is a testament not only to the oxygen-rich environment, but also to the evolutionary advantages of flight, combined with sheer scale, enabling stability in air currents and dominance over prey. Physiological adaptations were critical in enabling such gigantism. In millipedes like Arthropleura, robust jointed exoskeletons provided structural strength, counteracting gravitational forces. Their segmented bodies allowed flexibility and muscular coordination necessary for locomotion across the dense, uneven terrain of ancient forests. In flying insects, wing morphology and musculature evolved innovative designs that balanced lift, agility, and strength, accommodating their extraordinary size. The Carboniferous giants also interacted within complex ecosystems. Their feeding habits contributed to decomposition and nutrient recycling, influencing soil formation and plant community structure. Their presence affected predator-prey dynamics, occasionally prompting evolutionary arms races with vertebrates and other arthropods, further enriching biodiversity. However, gigantism was not indefinitely sustainable. As the Carboniferous gave way to the Permian, oxygen levels declined, climates shifted, and habitats transformed. These environmental changes likely imposed respiratory and ecological stresses that curtailed the growth advantages of large body sizes, leading to the gradual replacement of these arthropod giants by smaller, more versatile forms. While modern-day insects and millipedes are considerably smaller, the legacy of Carboniferous gigantism continues to inspire scientific inquiry into physiology, ecology, and evolutionary biology. It highlights the intricate interplay between environment and anatomy and exemplifies life's remarkable capacity for innovation. Imagine the primeval Carboniferous forests alive with massive dragonflies soaring effortlessly overhead and oversized millipedes rumbling across thick leaf litter. A world where scale and adaptation intertwined dramatically. This giant epic is a vivid reminder of life's extraordinary evolution. From these awe-inspiring giants, we now turn to the development of social behavior, the rise of early swarms and colonies that introduced cooperation and community living as powerful evolutionary strategies, setting the stage for complex animal societies before the age of dinosaurs. In the vast saga of life's evolution, one of the most compelling chapters is the emergence of social behaviors, early swarms, colonies, and cooperative interactions that transformed the dynamics of survival, reproduction, and ecological dominance. The development of social behavior among pre-dinosaur animals unfolded as a sophisticated strategy, enhancing resource utilization, defense, and reproductive success, fundamentally changing how groups of animals interacted and thrived. Social behavior, in essence, involves organisms interacting with each other in ways that benefit the group or individual members, ranging from loose aggregations to complex colonies with division of labor. The roots of such behaviors reach deeply into the Paleozoic era, where early arthropods, mollusks, and primitive vertebrates began displaying forms of aggregation and cooperation. Among the earliest manifestations was swarming behavior. Many Cambrian and Ordovician arthropods, including trilobites, exhibited evidence of synchronized movements in large numbers, likely driven by environmental stimuli such as spawning, feeding, or migration. Fossilized trackways and mass mortality beds capture these assemblages, revealing coordinated, repetitive patterns suggestive of collective action. Swarming offered numerous advantages: overwhelming predators through sheer numbers, enhancing mate finding, and improving environmental navigation. Colonial living took these interactions a step further. Sessile organisms such as early corals, bryozoans, and colonial algae formed intricate, self-supporting structures composed of genetically related individuals. These colonies exhibited division of labor, with specialized zooids or polyps performing distinct functions like feeding, reproduction, or defense. This specialization improved overall colony efficiency and survivability, allowing them to dominate reef building and other ecological niches. Social behavior also appeared in mobile fauna. Early eurypterids and primitive insects may have exhibited rudimentary social organization, such as cooperative hunting or group foraging. While direct evidence remains sparse, indirect indicators suggest that even simple communication and behavioral synchronization could enhance survival. Communication played a crucial role in social complexity. Chemical signals, pheromones, and other secretions likely mediated group cohesion, alarm responses, and reproductive coordination. Visual and tactile cues also emerged as components of social interactions, reinforcing group structures and cooperation. The evolution of sociality set remarkable evolutionary trajectories in motion. Groups could exploit resources inaccessible to solitary individuals, protect vulnerable life stages through collective defense, and respond adaptively to environmental challenges. Over time, social behavior fostered increased cognitive abilities, learning, and memory, a dynamic interplay reinforcing social complexity. Fossil records, modern phylogenetic studies, and behavioral observations converge to suggest that sociality, though ancient and often subtle in its early expression, played a pivotal role in shaping animal evolution prior to the age of dinosaurs. Visualize primordial seas and early terrestrial landscapes dotted with bustling aggregations. Worms writhing en masse beneath sediment, trilobite swarms traveling along ancient shorelines, and colonies of sessile animals fanning their feeding structures in synchronized array. These social behaviors echo as the origins of community, cooperation, and collective life that course through the veins of animal kingdom diversity. In the chapters ahead, we will witness how amphibians built upon these social and physiological foundations, evolving unique adaptations for their dual lives in water and on land, illustrating once again life's inexhaustible inventiveness before the rise of the dinosaurs. As life continued its trailblazing path from water to land, amphibians emerged as stunning innovations embodying a fascinating dual existence, a life tied intimately to both aquatic and terrestrial realms. These marvelous creatures evolved unique adaptations, allowing them to exploit diverse environments, overcoming physical and physiological challenges to thrive in habitats that neither fully aquatic nor fully terrestrial animals could dominate during their time. Amphibians trace their origins back to early tetrapods of the Devonian period, which had already developed limbs with digits, lungs for air breathing, and other features aiding terrestrial exploration. Yet their evolutionary success depended on maintaining strong connections to water, particularly for reproduction and early development, a constraint that shaped their distinctive life cycles and physiology. One of the hallmark traits of amphibians is their metamorphic life cycle. Beginning life as aquatic larvae, typically tadpoles, they breathe through external gills and inhabit freshwater environs. The transformation into air-breathing, four-limbed adults involves profound morphological and physiological changes. Internal lungs develop, limbs mature, tail structures regress, and sensory and nervous systems adapt for terrestrial challenges. This metamorphosis is remarkable not only for its biological complexity, but also for its evolutionary significance, exemplifying flexibility in coping with environmental transitions. Anatomically, amphibians evolved permeable skin that facilitates cutaneous respiration, absorption of oxygen directly through the skin, a vital supplementary respiratory pathway that supports their relatively low metabolic rates. However, this skin requires moist conditions to function effectively, anchoring amphibians to humid habitats near water sources. Physiological adaptations extend to osmoregulation, enabling amphibians to balance water retention and ionic concentrations despite exposure to fluctuating environmental conditions. Their kidneys and associated structures evolved to minimize dehydration and maintain internal chemical stability, critical traits for terrestrial survival. Behaviorally, amphibians often seek microhabitats that provide shelter, moisture, and feeding opportunities. Many are nocturnal to reduce water loss and avoid daytime heat extremes. Their reproductive behaviors typically involve laying eggs in water or damp environments, as their eggs lack hard shells and desiccate quickly. Sensory adaptations also underline their dual lives. Amphibians developed enhanced olfactory systems to detect terrestrial scents, and their auditory apparatus adapted to pick up airborne sounds, further augmenting their interaction with the environment. Ecologically, amphibians serve as vital connectors between aquatic and terrestrial ecosystems. Their larval stages participate in aquatic food webs, controlling algae or plankton populations, while adults often prey on insects and other invertebrates, regulating terrestrial invertebrate communities. Moreover, amphibians act as bioindicators, sensitive to environmental changes due to their permeable skin and aquatic ties. The Carboniferous and Permian periods were rich in amphibian diversity, ranging from small, agile species to large, salamander-like giants that echoed some aquatic ancestors' forms. This diversity reflects their successful exploitation of diverse niches and environmental conditions. Imagine dense, mist-laden river valleys and forested wetlands alive with the rhythmic choruses of amphibians, life cycles intertwined with water's ebb and flow, and with bodies perfectly tuned to traverse two worlds. Their existence represents evolution's poetic solution to the challenge of terrestrial conquest. As we ponder these amphibian marvels, our journey will next turn to a realm where predators sharpened their dominance, the Permian period, an epoch where formidable predators shaped ecosystems and set the evolutionary stage for the spectacular age of dinosaurs to come. As the Permian period dawned approximately 299 million years ago, life on Earth entered a transformative era that would sculpt the biological landscape long before the rise of the dinosaurs. This epoch, lasting until around 252 million years ago, witnessed the emergence and dominance of formidable predators that defined the age's ecosystems and set evolutionary precedents for future terrestrial and aquatic communities. Understanding the Permian predators illuminates how the foundations for complex predator-prey dynamics and ecological structures were laid well before the Mesozoic era. The Permian seas and lands teemed with a diverse array of predatory species, ranging from massive amphibians and reptiles to formidable synapsids, grouped as the mammal-like reptiles. These predators occupied multiple ecological niches, exhibiting varied hunting strategies, anatomical specializations, and life histories. Their dominance reflected ongoing arms races where prey species simultaneously advanced new defensive adaptations. Among the apex aquatic predators were large temnospondyle amphibians, possessing heavy skulls, sharp teeth, and powerful jaws suited for grasping and subduing prey. Their semi-aquatic lifestyles often mirrored those of modern crocodilians, patrolling freshwater systems with stealth and ambush tactics. Such predators thrived alongside primitive sharks and bony fishes, contributing to rich and dynamic aquatic food webs. On land, synapsids represented some of the most successful and influential predators and herbivores. Species like Dimetrodon, often mistakenly labeled as dinosaurs, sported large sail-like structures on their backs, possibly used for thermoregulation or display. Equipped with serrated teeth and muscular builds, these early synapsids hunted effectively in their terrestrial realms. Their evolutionary legacy is profound, as they bridge the gap between primitive reptiles and the eventual rise of mammals. Other predatory reptiles, such as early diapsids and pariasaurs, a group of herbivorous reptiles, contributed to complex community interactions, navigating a world of shifting climates and emerging biotic competition. The diversity of predatory strategies, from ambush, pursuit, to scavenging, demonstrated the adaptive creativity life was employing during this period. The Permian also saw environmental challenges, fluctuating climates, aridity, and habitat fragmentation that pressured predators and prey alike to refine physiological and behavioral adaptations. These pressures likely accelerated evolutionary innovations in locomotion, sensory function, and metabolism. Towards the close of the Permian, the largest mass extinction in Earth's history wrought profound changes. It wiped out a significant proportion of species, including many dominant predators, reshaping ecosystems and providing evolutionary opportunities for surviving lineages, eventually giving rise to the dinosaurs. Envision the Permian landscapes as theatrical stages where powerful predators ruled riverbanks, floodplains, and shallow seas. A world pulsing with the vigor of ancient life forms pushing biological boundaries. Their reign laid critical groundwork, ensuring life's complexity and resilience as it approached the dawn of the dinosaur era. As we move forward, the narratives of survival, adaptation, and renewal amid adversity guide us to explore how early animals weathered mass extinctions and built resilient legacies, creating the foundations upon which terrestrial life would flourish in the ages to follow. Throughout Earth's deep history, periods of flourishing biodiversity have been punctuated by catastrophic events of mass extinction, challenging life's persistence and resilience dramatically. These early mass extinctions, occurring well before dinosaurs walked the planet, tested the endurance of animal lineages and shaped evolutionary trajectories in profound ways. The story of surviving catastrophes reveals nature's remarkable ability to adapt, recover, and innovate amidst adversity, laying the groundwork for the diversity we witness today. One of the most significant early extinction events was the end Ordovician extinction, approximately 444 million years ago. Triggered largely by sudden and severe global cooling and widespread glaciation, this event decimated nearly 85% of marine species, affecting many early shelled invertebrates, reef builders, and planktonic organisms. Despite the scale of environmental upheaval, animals that survived demonstrated remarkable resilience, often by retreating to refugia, habitats where conditions remained hospitable, or by evolving physiological and reproductive strategies suited to fluctuating climates. The late Devonian extinction followed, sprawling over millions of years with multiple pulses, linked to global anoxia, oxygen depletion, climate shifts, and possibly asteroid impacts. This crisis severely impacted jawed fishes, reef ecosystems, and early tetrapods, yet it also spurred diversification among hardy groups, including amphibians and some fishes, which capitalized on vacant ecological niches to radiate and evolve new forms. Perhaps the most cataclysmic was the Permian-Triassic extinction, around 252 million years ago, the largest extinction event recorded. Known as the Great Dying, it eliminated up to 96% of marine species and around 70% of terrestrial vertebrate species. Swarming volcanic activity, massive greenhouse gas emissions, ocean acidification, and global temperature surges created a hostile world. Yet, in the aftermath, survivors from groups like some synapsids and amphibians laid the foundations for new evolutionary chapters, ultimately including the rise of the dinosaurs and mammals. Survival strategies during these extinction episodes often involved a combination of biological flexibility and habitat opportunism. Generalist feeders capable of exploiting a variety of resources often fared better than dietary specialists. Small body sizes, rapid reproduction cycles, and dispersal abilities also enhanced survival likelihood by enabling swift colonization of recovering environments. Resilience was also fostered by physiological adaptations. Tolerance to low oxygen, desiccation resistance, and metabolic versatility were traits that allowed survival in disturbed or marginal habitats. Some organisms entered dormant states or produced resistant life stages, like spores or cysts, bridging unfavorable periods. Fossil records from these extinction horizons often show rapid turnovers in species assemblages, highlighting the dynamic evolutionary experiments in recovery and adaptation. These periods of loss and renewal were crucibles that reshaped ecosystems, sometimes simplifying them initially, but eventually leading to more complex and stable communities. Nature's capacity to rebound was not limited to species alone, but extended to entire ecosystems. Early reefs, forest communities, and marine food webs saw phases of collapse and regeneration, emphasizing ecological resilience alongside evolutionary resilience. Picture ancient Earth enduring climate extremes, volcanic winters, and empty seascapes. Yet amid ruins, pockets of life persist, organisms clinging to existence, evolving new tricks, and over millions of years, repopulating the biosphere with renewed vigor and diversity. This enduring saga highlights evolutionary adaptability as one of life's most profound marvels. With the lessons of survival and resilience in mind, we now turn to consider the profound legacy of these pre-dinosaur adaptations, foundations upon which terrestrial and aquatic life would flourish in subsequent eras, ultimately shaping the vibrant natural world we inhabit today. As we draw our exploration of life's remarkable pre-dinosaur journey to a close, it becomes evident that the myriad adaptations forged over hundreds of millions of years provided an indispensable foundation for the flourishing of terrestrial life. Before dinosaurs came to dominate the Earth, countless creatures, through innovation, resilience, and evolutionary finesse, crafted the biological and ecological frameworks that sustained and nurtured life's onward march across the planet. The legacy of pre-dinosaur adaptations is woven deeply into every corner of modern ecosystems. From the earliest sensory breakthroughs, such as the evolution of complex eyes and sophisticated nervous systems, animals gained the perceptual tools needed to negotiate increasingly complex environments. These sensory innovations were crucial in shaping behavior, facilitating hunting, evasion, and intricate social interactions, setting the stage for the dynamic food webs that characterize terrestrial and aquatic worlds alike. Equally influential were the evolutionary developments in locomotion and structural protection. The mastery of movement exhibited by swimming pioneers like trilobites, and the innovation of protective shells and armor among various lineages dictated not only survival but also ecological relationships. These physical traits allowed animals to explore new niches, expand their range, and adapt to shifting environments with remarkable flexibility. The conquest of land by ancient arthropods and tetrapods, guided by the evolution of lungs, tracheal systems, and specialized limbs, represents a monumental leap in life's history. These adaptations overcame the challenges of desiccation, respiration, support against gravity, and locomotion on solid substrates, forging a path from water to the uncharted territories of dry land. They also fostered the rise of terrestrial food chains, introducing herbivory, predation, and symbiotic interactions in novel contexts. Moreover, early life's responses to catastrophes revealed the astonishing resilience and adaptability of nature. Surviving mass extinctions nudged evolution towards new possibilities, prompting diversification, experimentation, and ecological realignment. This resilience ensured continuity of life and seeded the diversity from which dinosaurs and eventually mammals, birds, and countless other forms arose. Chemical communication and social behaviors emerged as vital elements in the story, enabling cooperation, community building, and sophisticated reproductive and defensive strategies. These social innovations enriched ecosystems, enhancing survival through collective action and signaling the dawn of complex animal societies. This vast pre-dinosaur heritage offers us not only a glimpse into ancient worlds, but also profound insights into the processes shaping life's diversity. The biological legacies etched into fossils and genomes remind us that evolution is a continuum, a tapestry of adaptation, innovation, and survival across eons. As we reflect on this foundational legacy, we deepen our appreciation for the interconnectedness of life and the remarkable journey from simple multicellular organisms to the awe-inspiring diversity dominating today's Earth. This foundation paved the way for the extravagant radiations that would follow, first with the age of dinosaurs and later the rise of mammals, each chapter building upon the genetic, anatomical, and ecological groundwork laid long before. In essence, the pre-dinosaur adaptations were more than mere evolutionary milestones. They were the very blueprints of life's terrestrial success, resonating through time and space as testimony to life's enduring creativity, perseverance, and wonder.